So these are very common question. If you go and see the last 10 years, many of this have come. So self-directed learning is you have to know the answers, you have to know the points. Okay, since there are so many questions, at the end of the class we won't find time. So that's why I'll start discussing. So first question is bacterial capsule, then bacterial cell wall. You know bacterial, so sorry, first is bacterial capsule. So this is a question of pathogenesis. Okay, so morphology of bacteria and pathogenesis also, because capsule does both. So what you have to write, just see. So you have to first define that what is a bacterial capsule. So it's a layer of amorphous viscid substance around the outside, around the cell wall of the bacteria. So there are two things. One is a slime layer, one is a capsule. So when this layer is well organized and you cannot wash it off, it is called capsule. Okay. But if it is not, may I please know what is happening? This is not your drawing room, is it? Second last bench. If you're not interested, please get out. I don't have any issue. I'll take your attendance. You won't suffer. But don't disturb me. Okay, so bacterial capsule. It is an amorphous viscid substance around the cell wall, also called Glycocalyx. Okay, now Glycocalyx, if you can easily, if it is if you can easily wash it off, it is called a slime layer. But when it is well organized, you cannot remove it easily, then it is a bacterial capsule. Okay, so bacterial capsule plays a part in the pathogenesis of the bacteria because it protects the bacteria and also helps it to escape the host's immune mechanism. Another important point is most of the capsules are made of polysaccharide substance except Bacillus anthracis. So this is one exception. If bacterial capsule comes and in Viva voice also, the examiner may ask you what is the exception in the composition of bacterial capsule. So you will say Bacillus anthracis. Next is what are the functions? So obviously it gives the cell a form of protected rigidity. So protects from phagocytosis. As I told you, it plays a part in the pathogenesis. So helps in escaping the host immune response. So it prevents complement mediated bacterial lysis, prevents the cell from drying out. So in adverse condition also this will help the bacteria to survive, and protects from action of lysosome, bacteriophages. What is a bacteriophage? What is a bacteriophage? Viruses. Okay, so these are viruses infecting bacteria. Others, it's a source of nutrient and energy to the microbe. So again, helps in survival. Induces abscess formation, biofilm formation and adhesion. So helps in pathogenesis. In adverse condition, it will help to form the biofilm. Okay. And lastly, these are vaccine precursors. So many a times we see the capsular antigen is used for manufacturing vaccine. Example is pneumococcus, meningococcus and H. influenzae type B. These vaccines, you know, these are there. HIV, H. influenzae B, it is a part of national immunization schedule also. Okay. So for manufacturing these vaccines, we will need a bacterial capsule. Next, these are the examples. These are the types of bacterial capsule. This is the composition. So this I have highlighted. This is Bacillus anthracis. All are polysaccharide except Bacillus anthracis, which is polypeptide. Okay. Okay. Next. Now you will have to, next point is demonstration of capsule. How will you demonstrate the capsule? Negative staining, you all know what is negative staining. Negative staining by India ink. I know the example, but what is negative staining? What did ki hoy negative staining? Why do we call it negative? Yes. So background is stained in a dark, very dark color like blue or black, and the cells are usually not stained. The capsule is not stained, so you can see it remarkably. Next, serological test. This is a Quelling reaction. So this is done for visualizing the capsule of Streptococcus pneumoniae. Okay, so what happens here? You should know, at least you have to mention the name. Okay, so we will mix the antisera of Streptococcus pneumoniae with methylene blue stain. So methylene blue stain, when is mixed with antiserum and then the colonies are placed in it, the capsule will swell up and give this illusion of a round structure which we will call as Quelling reaction. Okay, so it's an illusion. Next is capsular antigen detection by serological tests like latex agglutination. Okay, latex agglutination, you all know we do. What are the examples of latex agglutination? Antigen antibody reaction. Any example of latex agglutination test? Yes? ASO, CRP, you don't remember? Okay. So similarly, there is, we can detect the capsular antigen by latex agglutination test for certain bacteria, certain microbes, such as Cryptococcus, it is a fungus, Neisseria meningitidis, and H. influenzae type B. So this much you have to write. So what are the points that we have to include in bacterial capsule? Just a recapitulation. Number one, about what is bacterial capsule, what is the composition, what are the functions. So composition, what is the exception in the composition, and lastly, how do you demonstrate capsule? Okay, this is one full answer. Doesn't matter if it comes in 5 marks also, you'll have to at least explain the headings. Okay, write 1 or 2 lines examples. And 10 marks mein aa gaya, so you'll have to write the whole thing. These points should be there. Okay, next question. Okay, next question is it short note on bacterial cell wall? Yes or no? You have the picture. Yeah. Okay. So first, again, we are following one structure, you see. So first we are defining, so it is, first is definition, it's a tough and rigid structure surrounding the bacterium. Then if you can remember, give the dimension, that is 10 to 24 nanometer thickness is there. Function, then you have to go to the function. So there are many functions. One and very foremost, first thing, it gives rigidity to the bacterium. Second is protection against osmotic lysis. Okay. Third is protect cell wall from toxic substances, again in bacterial pathogenesis. Site of action of several antibiotics. So this is for our help, that when we are giving some antibiotics, many of the antibiotics are acting upon the cell wall. Can you give me one example? Penicillin, very good. So penicillin is acting upon which enzyme or which part of the cell wall? Which protein? What is PBP? Okay. Now we come to it also, again, pathogenesis, it contains virulence factors, okay, like endotoxin. You know, there are 2 kinds of toxin, one is endotoxin, exotoxin. So bacterial cell wall contains endotoxins which contribute to the pathogenesis, and antibody against specific cell wall antigen, they provide immunity against bacterial infection also. So we have also used them in immunology. After function, you will then explain the structure of cell wall. Now we have two kinds of cell wall, gram positive and gram negative. Even I have kept it in details, at least know the heading, what you have to write. After definition, function, then structure. So structure is first, at least peptidoglycan, teichoic acid, you can write like this. Peptidoglycan is important because in gram-positive cell wall, this plays a part in gram-staining property of the bacterium. So this is very thick, about 50 to 100 layer thick than the gram-negative cell wall. Gram-negative has 2 to 10 layers. Here it is about 50 to 100 layers. And this is the composition of the peptidoglycan layer. If you can draw this picture, it will be very good. So you know the composition. So there are N-acetyl muramic acid and N-acetyl glucosamine. Okay, these two. And muramic acid will have side chains. What are the side chains? These are alanine, glutamine, lysine, and alanine. Okay, so these are different isotopes of the amino acid. These are amino acid side chains. So this side chain, that is L-lysine, will be cross-bridged with the last, that is D-alanine of another side chain. So this way, the whole peptidoglycan will be tightly cross-linked by the means of amino acid side chains and cross bridges, pentacycline cross bridges. Okay. So this structure, it gives the gram-positive cell wall a very unique quality. Can you please tell me that in gram stain, you see gram-positive cell wall as purple or blue and gram-negative as pink or red? Why is that? Why is that? Peptidoglycan. So what does gram-negative contain? Yes. So what happened is, I don't know if you get this explanation, in gram-positive cell wall, since there is this tightly cross-linked structure, so whenever you put acetone over it, that is a decolorizer, the pores, they shrink together. Okay, they shrink. Okay, so what happened is the primary stain, that is the crystal violet and the iodine, they form a complex, so they cannot get out. So that is why even when we are putting the decolorizer, the cell is not getting decolorized. So it is taking up the primary stain. But on the contrary, if you see gram-negative cell wall, it contains lipopolysaccharide. Lipopolysaccharide is it's like an oily substance. During COVID, you have done a lot of hand washing, so you must have seen now also we are in a habit of carrying the sterilizer or sanitizer with us, isn't it? So the sanitizer, it contains alcohol. Okay, it is also, we can say that it's a form of decolorizer again. So when you have oil or some oily substance on your hand and you put sanitizer over it, the oil gets removed, right? It gets dissolved. Same way, when you're putting acetone or alcohol over the lipopolysaccharide, it's getting dissolved. So whenever it's getting dissolved, the pore size is increasing and all the primary stain is escaping from the gram-negative cell wall. So what happened is, after that, when we are putting the secondary stain, that is safranin, it is taking off safranin. Am I clear? Are you understanding? So that is why this peptidoglycan layer is very important. So it gives the gram-positive its property of retaining the primary stain. So after that, we have teichoic acid. So after peptidoglycan, teichoic acid. This is again absent in gram-negative bacteria. This is also unique for gram-positive. So these are polymers of glycerol or ribitol, which are joined by phosphate groups. This maintains the structure of the cell wall. And this is not very, but there are two kinds of teichoic acid. One is cell wall, another is lipoteichoic acid. Next is gram-negative cell wall. So gram-negative cell wall, first let us see the structure. Actually, I should have put the picture on the previous slide. So this is the plasma membrane. Above, outside the plasma membrane, there is a periplasmic space, this much, okay? And from here, the cell wall is starting. This is the outer membrane protein, lipid A, and this is the lipopolysaccharide outside. Okay, this is a very thin layer of peptidoglycan is present here also, but it is very thin. Then the outer membrane protein and the lipopolysaccharide. So this is the composition of our gram-negative cell wall. So just at least if you can write the name that for peptidoglycan layer is also present here, but very thin, only 1 to 2 layers, and it is similar to gram-positive, but it is not cross-linked because pentaglycine bridges are absent. Then after that, what I told you, that is outer membrane protein. Outer membrane protein, it serves as a protective barrier and it contains some porine proteins. Okay, so this again helps in the transport of different substances. And last, this part, this is the lipopolysaccharide. This is the integral part. And this is the core polysaccharide, and it has O antigen or somatic antigen side chain. This is the O antigen. Okay, this plays a part in the pathogenesis. And periplasmic space is this. This is actually inside. After plasma membrane, periplasmic space, peptidoglycan, outer membrane proteins. This is a porine protein. Okay, and this is the lipopolysaccharide layer. Clear? So this much you have to write in cell wall. Clear? If you can draw the picture, if it's a 10 marks question, unlikely, but if it is there, you can draw the picture also. Okay, next, what was the next question? Next picture. Okay. So bacterial cell wall. Now a bacterial growth curve. So you must be knowing bacterial growth curve. Now what happens is, when a bacterium, it is inoculated in liquid media and after that it is incubated, so its growth takes a certain curve. Okay, first there will be no multiplication, then it will multiply rapidly, then there will be one stationary phase and finally a decline phase. Okay, so there are 4 phases of the bacterial growth curve. So first is this lag phase, lag, log, stationary, and finally phase of decline. It's very easy. You just have to write this definition, that when you can see it, how you can observe, and then what are the names of the phases. Next, just I have given it in every slide. You can draw one picture. Okay, this is very important. You have to give the picture here. So draw this picture, draw one picture, and below that you explain that is what is lag phase. This is the period of inoculation to beginning of multiplication. From here the curve is going up, right? So this phase is the lag phase. And here what happens, the bacteria, they increase in size, accumulate the nutrients so that they can multiply. So this is a preparation phase. So in this phase the bacterium will reach its maximum size, okay, and after that here it is the end of the lag phase. Then we come across the log phase. So log phase, there is exponential increase, okay, multiplication. So here the bacterium finally, since there is multiplication, so these are smaller in size, biochemically active, and here you can see these are uniformly stained. If you do the gram stain, they are uniform in staining. Next is the stationary phase. This is the stationary phase. So what is happening here? So the number of viable cells remain stationary as there is a balance between the dying cells and the newly formed cells. So whenever this log phase is increasing, obviously cells are also dying. Okay, here the rate of multiplication was higher. Okay, here what happening is dying cells and the newly formed cells, the rate of dying and newly forming cells are equal. So this is not the number of bacterium, it is about stationary. Okay, so there is a plateau phase or the stationary phase. So here again bacterium will become gram variable. Sporulation will occur. Why will sporulation occur here? When the spores form? Yes, it is obviously adverse condition is there. So there is some competition with the space and nutrients. So sporulation will occur and bacteria will produce some toxins, that is exotoxin and bacteriocins. This is to compete with the other bacterium. Okay. Next is the decline phase. Okay, this is the decline phase. So what will happen in decline phase? The cell death continues to increase. And what will happen when cell death is there? So because the nutrients that were formed here, okay, here and here, they will exhaust. Okay, so nutrients are getting exhausted, so more number of cell death is there. And accumulation of toxic products are also there. So that is why the bacterial count will decline and you will see involution forms. These are deformed cells. Okay. So this is pretty much all. So this will come as a short question. So you will write this part and draw the picture, please. Okay. Next we come to next question. Next. Yeah. Okay. So Koch's postulate. Who is Robert Koch? Who is he? A person, a thing, something? Yeah. Huh? At least call him a scientist, maybe, yeah? Okay. So, Koch's postulate, I've not put any introduction. Okay, if you can, because I doubt if you can remember this also. Okay, I see different kinds of things written. So, to be point, at least write what are the Koch's postulate. Okay. So, can somebody read this? Can somebody read this? Okay. So, number one is microorganisms should be constantly associated with the lesion of the disease. Okay. Two is it should be possible to isolate the organism in pure culture from the lesion of the disease. Third is the same disease must result in the isolated microorganism if it is inoculated in an experimental lab animal, and it should be possible to re-isolate the organism in pure culture from lesion produced in the experimental animal. And there is one fifth criteria which was added later on. So subsequently it was introduced. So it states that the antibody to the positive organism should be demonstrable in the patient serum. This is when serological tests were introduced. So this was also added. So if these four are okay, but this is only in your book only. I've not taken anything from outside. Now, exceptions, very important. So, there are some exceptions such as Mycobacterium leprae and Treponema pallidum, because it is said that it should be possible to isolate them, or it should be possible to grow them in suitable culture media, artificial. But these cannot be grown in vitro cultures. Okay, artificial media cannot be used. However, they can be cultivated in experimental animal in vivo. Then Neisseria gonorrhoeae, there is again experimental animal is not there. No experimental animal, however, it can be grown in vitro. So both is not possible. These are the two exceptions. As for your level, please at least write these. Okay, this you don't need. Okay. Okay, and one thing, this I have included. If you can write, there is one molecular Koch postulate also. So this was a modification. So it, at least this part, if you can write, all this you won't have to. So it states that the gene of a pathogenic microorganism that contributes to the disease should satisfy all the criteria Koch's postulate of the Koch's postulate rather than microorganism itself. Okay, this part at least. Okay, this is a molecular theory. Next is horizontal gene transfer. This you know better than me. So what are the different methods of horizontal gene transfer? Conjugation. First is conjugation or something else? Hmm? This is, can you see what is written here? Thank you. So first is transformation. So in transformation, you have to mention the Griffith's experiment. You won't have to be a very good painter for this. At least if you can write living S strain is virulent and the mouse dies because of that. Even if you cannot draw the picture, it's okay. But Griffith's experiment will have to be mentioned, okay? So what is transformation? What is transformation? By now you should be answering. Huh? Bolo? What is transformation? Transformation কি হয় ট্রান্সফরমেশন ছাড়া? কি বলছো? ওকে বলতে দাও না, ওকে জিজ্ঞেস করো। Yeah. Transduction or conjugation. Okay. So what is transformation? This is the easiest form actually. কি হয় transformation এ? Yes. Yeah. So again and again I'm telling you, please read the book because বইয়ের থেকে easily কোথাও দেওয়া নেই. It's very easily written and you can understand, you can grasp it. So transformation, it is a random uptake of free or naked DNA fragment from the surrounding media by a bacteria and incorporation of this DNA fragment into its chromosome in a heritable form. So এটার মানে কি হচ্ছে that, for example, there are two bacteria, one dies, so the genetic material from that dead bacteria are there in the surrounding, so this living bacteria is uptaking this naked DNA fragment and it is incorporating these uptaken DNA fragment into its own chromosome. Okay, this is very easily explained. And this was introduced, this was very accidentally, this was discovered by Griffith. So this is the Griffith experiment. So Griffith experiment ekhane ki hocche? Can somebody explain this? What is happening? Huh? Please come on. This was in school also, I think. Griffith experiment. Huh? Okay. At least what bacteria is being used here? Streptococcus pneumoniae. So there are two strains of Streptococcus pneumoniae. One is virulent, one is non-virulent. Okay. So virulent strain, it contains the capsule. Non-virulent strain is not capsulated. So virulent strain is due to the capsule, it is smooth. So we are calling it a smooth strain. And non-virulent does not have capsule, it is rough. You understand? So these are the rough strains. So usually when S strain is introduced into the mouse dies. R strain does not kill the mouse lives. Okay, in this living state. Now if you kill the S strain, okay, when the S strain is dead also, then also the mouse is living because it is dead. But when you are mixing the dead S strain with a living R strain. Okay, R strain does not have capsule. S strain has capsule, but it is dead. So the capsular gene or the genetic material is in the surrounding. So the R strain is taking that up and it is becoming capsulated and it is acting upon the mouse and killing the mouse. Okay, you understanding? So this is very easy. Just at least if you can write it, you won't have to draw it exactly like this. Just what is happening. This living S strain, mouse dies. Living R strain, mouse lives. You understand? So we are mixing of living R strain and dead S strain, mouse dies. Next we come to transduction. So what is transduction? At least from the picture. What is this virus? What is it? Huh? Bacteriophage. So transfer of the genetic materials by the means of bacteriophages. What is bacteriophage? This is a bacteria. This is the virus affecting the bacteria. This is the structure of the bacteriophage. Don't have to give. Okay, I just gave it to show you. So define at least write the definition. The transfer of portion of DNA from one bacterium to another by a bacteriophage. It's very easy to understand that when a bacteriophage is infecting one bacterium, it is transferring the genetic material into that bacterium. So this can get integrated into the hormone, into the chromosome, or it can stay outside. However, the genetic material is getting transferred. Okay. Now, there are two things. One is the lytic or virulent cycle. One is the lysogenic or temperate cycle, or called the lysogenic conversion. So lytic cycle की है that just look at this, that the bacteriophage is infecting the bacterium and inside the bacterium the chromosome it's getting fragmented into different parts later on. Since the genetic material of the phage is inside, so they will multiply. Okay, they will multiply into different parts of the phage. Later on, so one will have the head of the phage, one will have the body of the phage, all these things. So all these proteins are getting formed inside the bacterium. Later on, there is assembly and packaging, okay, into different small phages. So when this packaging is happening due to faulty packaging, that is sometimes what happens inside this, you will remember the bacteria's own chromosome was also there. So sometimes the phage, what happens during the packaging, it is taking up the bacterial chromosome also inside it. Okay, and finally what happens, this cell, it tears apart, ruptures, all the phages are released and they will go and infect another bacterium. So any of the phage that will contain the bacterial chromosome or genetic material, it will again transfer this to another bacterium. Okay. And this is about lytic cycle. Lytic cycle can also karo. Bacteria is getting lysed. Okay, bacteria is dying. Another is the lysogenic cycle. So in contrast, lysogenic cycle ki hoy, the bacteria lives. Okay, nothing happens. Do we have the picture? Okay, let me just explain this. The lysogenic cycle, what's happening is the genetic material is getting in and it is getting integrated with the chromosome. It is remaining like that. Okay, multiplication and packaging, rupture of cell, these are not happening. Now I have kept this picture just to show you. So this is a bacteriophage. This is the genetic material from the bacteriophage. This is the bacterial chromosome that was there already in the bacteria. Now all of them are getting fragmented like this, protein are being produced, and during the packaging you see this phage is taking up the bacterial chromosome, and this is again infecting other cells. So three things can happen. One is either there is survival of the donor DNA. Okay, so when this phage is infecting another bacterium, so three things can happen. What is the fate of transduction? So either this donor DNA, this can remain separate outside the chromosome. Another thing is unstable gene transfer. The phage may have transferred the gene, but inside this there is fragmentation and disintegration of the genetic material. Third is a stable gene transfer, that is, it is getting incorporated in the chromosome and it is there as a heritable chromosome part. Okay. This is the same thing. Okay. Now, next is conjugation. Okay. So, in transformation, and why you need to write this in order, because in transformation and transduction, the both are happening when the bacterium are apart. Okay, so there is no contact between the bacterium, but conjugation, the two cells are coming together. Okay, they are in contact, one conjugation tube is being formed. So um, so there are several types of conjugation. At least if you can write few forms, one is the F plus F prime and F plus F minus form. One is the high frequency conjugation. Then the R plasmid, that is resistance. I'll just show you what is the F plus F minus. So F plus is the donor bacteria. It is also called the male bacteria. So what will happen? This contains a F plasmid. This is the F plasmid. Okay, outside the male chromosome. So this F plasmid is getting transferred to the F minus cell. Okay, and by the form of conjugation tube, and subsequently the F minus cell is becoming F plus. Another is the HFR. HFR is the high frequency transfer of genetic materials. So this, what happening is, this is integrated inside the chromosome. Okay, so this is by conjugation, a part of this is getting transferred to another bacterium, but here this does not become F plus, it remains F minus, because during the transfer halfway the tube will break. Okay, yes, what is so funny? Define transduction. What is transduction? Yes, two girls. What is transduction? At least tell me, I have taught you. At least have the courtesy to stand up and tell something. Next is resistance factor. So, conjugation, F plus, then HFR, and at least this one you can write, this is resistance factor F, because conjugation is a mean of transferring resistance to antibiotic, resistance gene. Okay, so transferable drug resistance occurs by conjugation, one of the means. So what happening is here, that there is one like F factor, this is R factor. Why R factor? Because this contains the different resistance genes for the different antibiotics. So by one cycle of conjugation, many drug resistance genes can get transferred. So we get a lot of MDRs. What is MDR? multi-drug resistance. So how does this happen? So you will see many a times we are due to the misuse of antibiotics. So overuse of antibiotics. Initially the patient was responding, later on the patient is not responding to a certain antibiotic or maybe a group of antibiotics, more than 3 drugs in one class. Why is this happening? That initially there may be one bacterium or one microbe that was resistant. Later on, due to conjugation, that is due to our plasmid transfer. That is, in one plasmid there are many determinants, many small genes. So those will be transferred, and at one go, this other bacterium also will become drug resistant to all the antibiotics that we are prescribing. Okay. Cholecynogenic factor, this you just know, these are bacteriocins. Okay, bacteriocins are, these are like antibiotics, but these are produced by one bacterium to inhibit another strain of bacterium. Okay, so bacteriocins are also transferred by conjugation. So conjugation are doing, there are two things at least you write, the transfer of drug resistant genes, resistance, and another is bacteriocins or cholecynogenic factor. Both are same. Okay. Next is sterilization. Not going into any details. Write the definition. What is sterilization? Tell me fast. Sterilization is? Bolo? Just read and tell me. It is process by which all living organisms, emphasize, all living organisms including the viable spores are either destroyed or removed from an article, body surface or medium. Every word is so important. Every word. Okay. All living organisms including viable spores either destroyed or removed from articles, body surface or medium. This is the exact definition, you need to write this. And this results in reduction of 10 to the power 6 log colony forming unit. These two things have to write. Next, disinfection. Disinfection is, it's a process to destroy or remove most, if not all, pathogenic organism but not bacterial spore. So what does this mean? That in disinfection we are reducing the number of microbes to a level where it is not pathogenic. Okay, for the usual course rounds and all, we are using disinfectants. Okay, but when we are preparing media, for example, we have to sterilize them, or we are using some instrument in the OT. Okay, that is very highly critical instruments. Okay, invasive instruments. We have to sterilize them. So this is reducing 10 to the power 3 log colony forming units of microorganisms. Next, the table. So you understand two definitions, how many log colony forming units are getting reduced, and then the this table. This table is that physical methods and chemical methods. Two types of sterilization, disinfection are there. Physical methods, drying and heat, not regularly used in healthcare settings. We are using dry heat and moist heat. Very important. Dry heat is flaming incineration, hot air oven. Out of this, have you seen incinerator? Have you seen incinerator? Okay. So flaming, you know what is flaming? In Bunsen burner, you are flaming the mouth of the test tube. Okay. Red heat you are providing to sterilize our the straight wires and loops. Next, incineration. Incineration is usually done in waste management. Okay, biomedical waste management. So these wastes are incinerated or combusted at very high temperature, like 1200 degrees Celsius, 1600 degrees Celsius. And lastly, hot air oven. This will come again, principle of hot air oven. This is another question. So you have to write the temperature and time. So what is the temperature and suitable time for hot air oven? How many hours? In hot air oven, what are the articles that we are sterilizing? Hot air oven e ki sterilize kora hobe? Please start studying, please. Yeah. Okay. Now moist heat. In hot air oven, you sterilize the glasswares and paraffin oil, and the time is 160 degrees Celsius for 2 hours. Next, moist heat. Moist heat is, now moist heat you have to divide temperature below 100 degree, temperature at 100 degree, temperature above 100 degree. Below 100 degrees, pasteurization, water bath and inspissation. Temperature at 100 degree, you all know, boiling, steaming, tyndallization. And temperature above 100 degree, very important, autoclave. Autoclave principle is very important. You have to read the principle. Hot air oven and autoclave, these two principles. Even if you can't write anything, at least the definition and the principles. Okay. Next is filtration. Filtration, depth filters, membrane filters, the HEPA filters that we see in the OT, the biosafety cabinets, all these things. Next, radiation. Radiation can be ionizing radiation or non-ionizing radiation. One example. Ionizing radiation is gamma rays, X-rays, cosmic rays. Non-ionizing, ultraviolet and infrared. Last, ultrasonic vibration. This table is for the physical methods. Next, chemical methods, chemical sterilizers or chemical disinfectants. Those chemical sterilants, if you know the groups, if you know the classes, that is also enough. Another is the Spalding classification. I have not put it here because the question was not set like that, but another thing is the Spalding's classification of sterilization and disinfection. Write it down, you forget. Spalding's classification, read it. Chemical methods, alcohol, aldehyde, phenolic compounds, bipionides, halogens, oxidizing agents, very important, hydrogen peroxide, okay, in plasma sterilization. Oxidizing, then heavy metals like mercuric and copper salts. Surface active agents like quaternary ammonium compounds and the soaps. Aniline dyes and gas sterilization, again very important. So low temperature steam formaldehyde, ethylene oxide, very important for the machines, lung heart machine. Then beta propiolactone, plasma sterilization, again we are using the gaseous form and some ions in plasma sterilization, so we are activating them with electricity, electric current. So more important is the physical methods. Don't leave anything over here. Chemical methods, even if you forget some of the points, it's okay, but at least write the classes and mention like these are the sterilants, these are the chemical sterilants, and this is gas sterilization methods. Okay. So along with this principle of either autoclave or hot air oven, or maybe if you are not that lucky, maybe what is inspissation or what are the uses of tyndallization, like that can come. So here I am just, this is for example only. So if you can draw the structure of autoclave, that will be good. Otherwise you just write the principle. Can somebody tell me the principle? Autoclave, what is happening here? First of all, this is a method of moist heat sterilization above 100 degrees Celsius, and we are using steam as it expands and has a higher temperature than water. So when steam is coming in contact with cooler substances, it is condensing or it is getting sucked in and it is releasing a huge amount of heat. What is the heat? That is 1600 ml of steam. It condenses into 1 ml of water at 100 degrees Celsius and releases 518 calories of heat. If you just tell that it releases heat, that is okay. So in this heat, normally most of the viable spores are also destroyed. So this is the principle. Okay. Along with that, what is the temperature and pressure and time for autoclave? How are we increasing the temperature? By increasing the pressure. Okay. So, yes, what is the temperature? So these are the conditions. So most commonly used is the 121 degrees Celsius for 15 minutes at 15 pounds LPS or 15 PSI, pounds per square inches. It is the most commonly used sterilization condition. Also there are others like 126 for 10 minutes at 20 PSI. At least this you remember, this part. Okay. Okay. We are going to the next. What is the time? Okay. So next is normal microbiota. Actually, I have clubbed two questions together. Either distribution can come, another role. What is the, just one short note can come. You have to write at least point wise. You have to write what is normal human microbiota. Then there are two categories: resident and transient flora. And then one example if you can give, that is E. coli is a resident flora of the intestine. They do not cause harm, but they are beneficial. But the same E. coli, it can cause harm if it is introduced in another region. Okay. These are the beneficial effects, then the harmful effects. Beneficial effects again prevents colonization, synthesize vitamins, immune stimulation, complement activation, all this. Harmful effects. So these are in immunocompromised patients. So even the normal microbiome in immunocompromised or post-transplant cases can give rise to infection. So that is one. Another is, one beneficial effect is obviously it reduces the infection. I forgot to say. So waste produce antagonize other bacteria. Just go and read. I don't have time. Okay. So another, I just told you that harmful effect if they enter a wrong site or tissue. That is, if it is a sterile like blood. Okay, blood is sterile. There is no microorganisms, but somehow during some procedure or something, this normal microbiota, normal microbial flora gets introduced into the blood. So it will cause infection. Okay. Now, anatomical site distribution. I have given two questions. One is the distribution, another is the role. So role I have explained. Distribution is, now anatomical site wise you will divide the distribution. Okay. This is in your book again. Just see. No need to capture picture. Book hai achhe ka. So you will just at least, if you can write the predominant flora, that is in saliva or the buccal cavity gingiva, predominant microorganism is Viridin streptococci. Okay. When there are more anaerobic cocci also, but predominant is Viridins. Next is the nasopharynx. Nasopharynx again, nasopharynx and these are all mouth buccal flora as well as the nasopharynx. So the flora will overlap. Again, streptococci, alpha, non-hemolytic, Neisseria, a non-pathogenic species, diphtheroids, very important, and cons, that is Staphylococcus epidermidis. Okay. Then GIT, gastrointestinal tract. So in gastrointestinal tract, very important, you will have to explain the anaerobic flora. Anaerobic cocci is there. And Helicobacter pylori. Where is Helicobacter pylori found? Does Helicobacter pylori have any pathogenic effect? I'm just asking. Okay. So, yes, Helicobacter pylori, which disease is associated? Last bench. Good. So, peptic ulcer disease, which can lead to gastric carcinoma. Now, predominant are these, including enterococci and streptococci and Streptococcus agalactiae. Next, we come to the female genital tract. Predominant are Corynebacterium species, not diphtheria species. Okay, so don't write any pathogenic bacteria here. Lactobacillus species, very important. Okay, so Lactobacillus species, there is one score that is Nugent's score you see. So Lactobacillus species should be predominant, should be abundant. If it is less in number, that means that this will give space to the pathogens. So pathogens can colonize and cause infection. Next, next is obviously skin. Now, most important is Staphylococcus epidermidis. Please remember, and diphtheroids. These two should not be missed. Okay. Others are obviously Streptococci neisseria are there. There's some fungi also. In skin, anything can come you can come across, but these are important. That is Staphylococcus epidermidis, diphtheroids, and some anaerobes like Propionibacterium. So Propionibacterium is again, it is associated with acne vulgaris. Okay. When the bacterial load will increase or you're immunocompromise something. So okay, this much you can write. So role, another thing, this is not included. Another thing is the prebiotics and the probiotics. What does probiotics do? What does prebiotics do? Write it down. These are also potential questions. At least definition. What is probiotic? What is prebiotic? Okay. Now next. Okay. Next is antimicrobial resistance. So mechanism of antimicrobial resistance. Another question can come, that is what are the classes of antimicrobials. So there is an enormous chart for that. You will also read them in pharmacology. Classes of antibiotics, what are the classes, what do they do, how they act, okay. But obviously in our part we will teach you exclusively the mechanism of antimicrobial resistance. So how any microbe, it acquires the resistance. So one is that, what are the mechanisms? One is that decreased permeability across cell wall. So cell wall may be too rigid, permeability is less, so the antimicrobial is not getting penetrated. Okay. Examples: Pseudomonas, Enterobacter and Klebsiella species against drugs such as imipenem. Okay. Aminoglycoside and quinolones, fluoroquinolones. Number 2 is efflux pump. Efflux pump, what is happening? That is, maybe the antimicrobial is penetrating into the bacteria, but due to the activated efflux pumps, they are getting expelled. Okay. So expulsion of the drug from the cell. Example again, E. coli and other Enterobacteriaceae, again tetracyclines and chloramphenicol. Staphylococcus also against fluoroquinolones. Examples. So this does not have any meaning without any example. So give one example at least. Okay, third is that enzymatic inactivation. So the bacteria, it is producing, or the microbe is producing some enzymes that will inactivate the antimicrobial property. Now these are the some enzymes, that is beta lactam is very common, you know. This inactivates the penicillins. Okay. Next is aminoglycoside modifying enzyme. Structure of, again, aminoglycoside modifying enzyme, you know whom it is inhibiting, aminoglycosides. Chloramphenicol acetyl transferase, again chloramphenicol. Only beta lactam is, it breaks down the beta lactam rings. Okay, beta lactam ring is in penicillin. Cephalosporins also, you know, beta lactam ring is there. Yeah. So all the drugs which, all the antibiotics or antimicrobials containing the beta lactam rings are getting inactivated by beta lactamase. So what we are using a beta lactamase inhibitor. Okay, BHI with that. Sometimes there is ESBL, extended spectrum beta lactamase. So there we will need broad spectrum antibiotics for that. Okay, all these are examples, enzymatic inactivation. Next is modifying the target sites. I gave you one example, that is Staphylococcus aureus, PBP. Okay, there is one thing, there is methicillin resistant Staph aureus. What is methicillin resistance? Do we use methicillin? Have you? No, we don't use. But these are, it means that these are resistant to all the penicillin class. Why is it happening? Because the target site of penicillin is penicillin binding protein in the cell wall. Okay, but what is happening here? MRSA, in methicillin resistant Staphylococcus aureus, this PBP is getting transformed to PBP2A. That is, there is modification of the target site. Target site is getting modified. So the antibiotic, it does not recognize the PBP2A and does not get attached, does not work. Also rifampicin resistance in MTB, very common nowadays, RIF resistance. So that is also because of modified target sites. Okay. Then we come to antimicrobial susceptibility test. Antimicrobial susceptibility test. Okay. Just listen to me. When you are writing antimicrobial susceptibility test, you can give it in form of a flow chart or a table that what are the different types of antimicrobial susceptibility test. Okay, here I have given it in writing. You can form a flow chart also. How will you do it? So you will write first antimicrobial susceptibility testing, then you will under it you will give the diffusion methods, dilution method and automated methods. Diffusion and dilution, these are manual methods. Okay, these are conventional methods. In diffusion you will write the different examples. Examples, that is Kirby-Bar disc diffusion, Stokes diffusion, or this E-test, epsilometer test. That, do we have a picture? Oh, we don't have a picture. Epsilometer test is also disc diffusion, but it is one gradient is there around which the inhibition zone is there. So we see the MIC value in that. This come under disc diffusion. Then the dilution methods. Dilution method mein there is broth dilution, liquid dilution, then agar dilution methods. And in all these methods, let me tell you, we calculate the MIC. What is MIC? MIC is minimum inhibitory concentration. What do you mean by minimum inhibitory concentration? Anything, any crazy idea? What is minimum inhibitory concentration? Okay, so MIC is, another thing is MBC, but this is more important because most of the times we are calculating the MIC. So MIC is that it is the lowest concentration of drug, concentration of antimicrobial at which the microbe is not growing, visibly it is not there. Okay, so that is the MIC, lowest concentration of antibiotic at which we cannot see the bacterial growth. Okay, minimum inhibitory concentration, minimum concentration of antibiotic. If it is more, that means it is resistant to that drug. Okay, so in all these methods we are calculating the MIC. In these method also, epsilometer test also, we are calculating the MIC. Only Stokes and Kirby-Bar disc diffusion, we are calculating the zone of inhibition. So these are the zone of inhibition. We do a lawn culture, carpeting lawn culture of the colony, after which we put the antibiotic discs like that and we incubate it in the incubator 37 degrees Celsius overnight. Next morning we will calculate the zone. Okay, from the center to we will calculate the highest diameter of the zone and then we will compare them with the CLSI or UCAST guidelines. Okay, so what does these guidelines do? They give us the susceptible, intermediate and resistant values, the zones. Okay, so below this zone it is resistant, or it is intermediate or susceptible. Well, another discussion can be what is susceptible, what is intermediate, what is resistant. You just know susceptible is clinical, it is effective in vitro. In immediate, it can be effective, it is, but at higher concentration, and resistant, even if you increase the concentration of drug, it won't be effective. Okay. Last is the automated method. Automated method only. This also calculates the MIC value only, but this occurs in a machine. So you will have to give the examples, that is Lytic, Microscan, Phoenix. Okay, these are using the broth dilution or agar dilution methods, but these are in a machine, so manual labor is less. So large batches can be prepared and the susceptibility test can be done, less time consuming. Okay, last is lab diagnosis of fungal. Just bear with me 2 minutes. Lab diagnosis of fungal disease. Now, as you know, nowadays in general microbiology, general bacteriology, general parasitology, and general mycology is also there. Okay. Bacteriology we will read, parasitology different. We have done stool examination, all that you have gone through. But one question can come specifically this, that lab diagnosis of the fungal disease, or what are the different fungal stains used, what are the methods by which you do culture. Okay, so whenever this comes you will have to know the sequence. That is why I have given it. Now because fungal infection can happen everywhere. Okay, it is, there are different, the skin and submucosa, then the systemic fungal infections. Okay, now specimen, first you will say specimen collection. It depends on the site of infection. Then we will do all these things. What are these things? That is, we will do the stains, that is microscopy by KOH mount and gram staining. And India ink or nitrosin are negative stain, but there are some fungus which are capsulated, like Cryptococcus. Okay, it's a capsulated microbe, so we can visualize the capsules. Then histopathological stain, and lastly after culture you can do LPCB. Another question, very important, what is LPCB? Full form, lactophenol cotton blue. But this does not end here. Lactophenol cotton blue, you will have to know the function of each component. What is the component of lactate? What is the component of phenol? Cotton blue. Glycerol. Glycerol is also there. Okay. Can anybody tell me what are the functions? So you have to know the function. Okay. Okay. Next is the culture method. So that was microscopy. After that culture you will have to write the culture media. That is most common is the Saboros dextrose agar, neutral SDA. These are the corn meal agar and rice starch agar. These are nutrient deficient media. Chrome agar for candida. So this is the example of chrome agar. See, so candida, when we are suspecting candida disease, we can inoculate like this. So different colors of candida species will come. So according to the color, like if it is like this, if it is white, it is albicans. If it is bright blue, okay, you will see this is tropicalis. Next, culture identification. Then after culture, you do the identification. First thing you do is you do a LPCB mount, okay, or gram staining in case of yeast. Another thing, culture, you have to incubate at 2 temperatures. One is 37 degrees, one is at room temperature, 25 degrees Celsius. Why is that? Because dimorphic fungi can also occur. Dimorphic fungi grows in 2 temperatures. So both have to be covered here. Then culture identification, we have to see first you have to examine the culture. Okay, culture plate, that is you have to see the rate of growth, pigmentation, if there is any texture. Okay, rate of growth, very fast growing, like some molds are very fast growing. So within a week or something. Dermatophytes are very slow growing. So about 3 weeks you will have to see. Okay. Next, you'll have to give a culture like this. You can do a slide culture, teased mount. You can tease it and do a LPCB mount, see it under the microscope. Okay, these are the different other methods are also there. For candida, some specific tests are there like germ tube, dull mount plate, carbohydrate, fermentation test. Dermatophytes, from the specimen, we can do a hair perforation test or dermatophyte test media. Okay.